hope it was also lively discussion around. And what we would like now is, so we had 5 groups. If one rapporteur or someone willing to share a bit of feedback on the 2 questions, that would be great. And we could, the plan is to do a brief summary of this workshop and perhaps to come up with some actions. Well, in my group, we were even thinking about a book or something on protocol, because it came across maybe at national level, there is some methodology. They are standard, but not used in different countries. So maybe, and it's, to our knowledge, there is no centralized book like a protocol, or like where I was used to do with the Environment Agency, like a blue book recommendation of methods, and where we all are agree and argue. Maybe there is no single methods, and therefore harmonization will be difficult. By having methods with the pro and the cons or limitation, or what is the intended use of the method for measuring what, would be useful to be collected. So one thing also we were potentially thinking via the website, we could point out or give link on whether we are aware of protocol they are approved and used per country. So at least it will help to share information. So I'm already stepping on summarizing what the group has said. Is anyone would like to start to share maybe views on the what is needed for sampling and analysis methodology, and maybe going towards harmonization?
I'll kick off if you like,
yeah.
Fred. Just give you a very quick one. We had a very small but perfectly formed group, and I'm not sure we really answered your questions, but I'll tell you what we talked about. We talked quite a bit about the need for silos to be broken down. So we were just trying to sort of look at the methodologies that might be used across the indoors and outdoors, across chemistry and biology. We even talked about sort of fundamental science versus applied science as well, and how within the network. So all of these specific areas have got their own methodologies, some of them standardized, some of them not, but they don't cross-fertilize very well. And so that's why the network's so important. So it's about sort of methodology. It's not necessarily harmonization, but it's the sort of the complementarity of methods came across as being very important. And I think Carla was making a point in her presentation about how mixed methods, combining methods together, gives you a rich picture of a particular environment. And so I think that as a theme came across from our group. And what one of the scientists in my group is looking at indoor air, and effectively he's bringing lots and lots of kit into a controlled environment, you know, different types of measurement devices, including low-cost sensors, to get a sense of what is in the air and how these different measures correlate to each other. So I thought that was a really exciting idea. And also we had people who are working at the real sort of fundamental levels of the viability of bioaerosols and environmental impacts on viability, and how that kind of fundamental science is so essential when it's complemented with the kinds of applied science that we might do in the context of, you know, outdoor air and emissions from industrial facilities, or from the effects of people's behaviors like cooking or cleaning on air. I suppose the other main thing we talked about was low-cost sensing, and where we got to on that with biological sensing. And really, I think the answer's not that far. But going back to the points that Rob Kinnersley made in his presentation, I think there was in our group there was a lot of enthusiasm for low-cost sensing, even though it might have its challenges in terms of variability and precision because of just how valuable that temporal data is in homing in on emission events, whether you're indoors or outdoors, and that being the indicator and the focus of them being able to go in with a more sort of detailed specific reference method for which we have more confidence in that. So big thumbs up for low cost sensing and looking at the ways in which it can be of value. And I think maybe the last one was just that we wondered whether or not there was you know, equally good indoor and outdoor air quality monitoring kit for PM and biological PM, and whether or not there might not be quite such a, you know, a good range of options for indoor air. We weren't sure, but we felt that might be the case. I think that was it for us.
Right. Next.
We go next, Brad.
All right.
So we had a great discussion in our group, but like with your workshop breakout room, Fred, Rob suggested a key output of the BioAirNet should be some kind of compendium of measuring systems. So looking at the pros and cons of each method, looking at things like portability as well, and costs and efficiency and what the method does. So yes, so I think that should be one of the key outputs of this network. And then other things that we came up in the discussion, not necessarily related to the questions, but these were the points that we sort of discussed, that there's a need for a specific protocol from the lab to the field, and it depends on the industry, so it has to be industry specific. What might work for one sector might not work for another. We need to know the complete composition of the bioaerosol, and which fractions of that complete community are the ones that cause the human health. So I guess not just the composition, but also the relative abundances of those as well. And then we need to think about a suite of target organisms that we can look at. And again, this depends on the industry, so it would be specific for a particular industry. And then another thing that was raised is perhaps we need to have, as a more routine type of measure, collecting size fractions and surface sampling in parallel, so that we can look at the inhalable fractions that cause health effects. So looking at like workplace exposure. So those were the main things, Fred. I've got them written down so I can send them to you if you missed anything.
Yeah, that's great. Zaire?
All right, yeah. We had a sort of a very vibrant group coming from Environment Agency and industrial partners as well. So basically, just more or less the similar things that Sean and Corinne has highlighted. But regarding the question one, for some sort of challenges are how can we bring some sort of methodology harmonization. I think the one point that came out in loads of discussion was this, that before even to do this, we need to have some sort of confidence or identification of the endpoint, which endpoint we are interested in terms of our measurement. And if we have some sort of common endpoints and we identify them, then it's easy for us to come up with some sort of a framework of different methods, you know, and then we can compare them basing or focusing on that specific health endpoint. Another thing that regarding this was also highlighted was this, that at the moment there are loads of methods, okay, and each of that method can give you independent information. So based on those sort of individual methods, perhaps there are good sort of standardized let's say approaches available for that specific method. But is it possible for us to have, let's say, a kind of a decision tree analysis where we can see that what are the different methods, sort of standardized approaches for individual methods, and then building on that one to coming up some sort of commonalities regarding once we have identified that endpoint in terms of our specific analyte. So also for specifically, there was quite a lot of discussion on real-time detection using UV lift sort of devices as well, because Ernie was there as well. So for that sort of side of the things, we still believe that there is a big work need to be done in terms of harmonization with regarding to have some sort of a reference materials for fluorescent signal lifetime, as well as to assigning that sort of a fluorescent signal back to the origin of a specific biological material. And for question number 2, the key thing again that was highlighted, that there is a big role for emerging technologies to give us a better understanding of the fundamental questions about, let's say, the kind of biological material that we have in different environments. But even to translate that knowledge to regulate, and is the main thing is to have more understanding on the toxicology, because that was the point that was highlighted, that we don't have enough sort of data to relate those sort of information that we are getting utilizing these different technologies to the public health exposure. So probably that is a way forward that we should focus getting more information or trying to link with the colleagues from the health if we really want to have some sort of impact of translating this knowledge back to the regulations, be in sort of outdoor environment or indoor environments. So that's
Okay.
it.
Great. Emma, you are the they put you all volunteer as well?
Yes. Well, there was only 4 of us, including me. So yeah, so I let them off. And we were predominantly from sort of research background, I suppose, but we covered quite a lot of the same things, particularly with Sean. We had that idea that there's a balance between using different methods to have a more holistic view of what you have there, versus if you know there's something in particular you're interested in, you know, you might want a more specific method just to look at that. We thought about sort of, I guess, the harmonisation more in terms of a research perspective, maybe less so than a regulatory point of view. And we talked about how it might be useful to have publications on methods for sampling analysis, not just what instruments are used, but how and where they're used, what parameters, you know, sort of if you're sampling, what locations should you sample, to kind of help researchers make more informed choices based on their research question, especially for those that were new to the field or less experienced. And I guess the flip side of that is the challenge we sort of, or challenges we highlighted there, was that this sort of information needs to be kept up to date with a view to question 2. And, you know, Fred, you mentioned maybe having some like a website somewhere, but how do you encourage a large number of people from different backgrounds to read that and actually take up the sort of recommendations or make those informed choices? And then we sort of talked about whether you'd need different considerations for different types of bioaerosols and things like that. So there was quite a lot of discussion there. And then for question 2, I think we could all appreciate there was really great opportunity to advance characterization with a huge range of different new methods and technologies. And it's not just the composition, but we had a PhD student who's going to start looking at how bioaerosols behave when they're inhaled, and, you know, what effect humidity has on them and things like that, and therefore where they end up in the respiratory tract, because that's going to be really key as well for assessing risk to health if you're thinking about regulation in terms of the human health side rather than the regulation in terms of measuring emissions from different types of sites. So, and like Zahir said, that feeds really nicely into theme 3 when you're thinking about the health impacts of bioaerosols. We also sort of said if you want methods to be taken up by regulatory bodies or to be used to inform regulation, then you need that comparison to the existing methods to show what benefit they're providing, what improvements they're providing. And then someone in the group had a really nice link already, a relationship with industry, and it sort of highlighted how important it is to build those relationships with industry so that they were, you know, taking on board what research was coming out. They were providing suggestions to inform the company, and they were making changes to their risk assessment. So it's really allowing you that cycle of trying things out and developing, you know, what works best. And we thought, you know, having those relationships was really key to actually impacting on regulatory needs. So yeah, that's where we got to.
Great stuff. Well, in our group, quite a lot of been already said. I think what I come across also is how do you define a healthy microbiome? We all agree we are very good at characterizing, but exactly what that means when you characterize and what make a healthy microbiome and how you relate these to a health. And we felt also perhaps where the emerging technology, you do have air quality just on NOx, CO, or UV index. You don't have a air quality index for microbiome, your microbiome, and how that might affect vulnerability or asthma. We do have some kind of already for pollen, but not so much for the others. And we felt perhaps opportunity to get molecular technique integrated with biosensor and sensing the environment might be where novel technology would be helpful, perhaps for mitigation or inform future policies. We also felt where the novel technology could play a role is on background, and that refer back to what Rob was mentioning, because interestingly for pollutants, there's been a lot lobby from the industry to make sure you get regulated for what is above background. You don't want to be blamed for something that, you know, if you, and for the biological aspect is not something we came yet across, but we don't have much background level. So we don't know what might constitute an environment more, or might be triggering some response versus another. So a call for maybe some national background monitoring network would be potentially where novel technology could also contribute to. And we back to, and on the health indicator, and we had quite a nice chat with Graham, where effectively within the life there is we can be exposed to different bioaerosols, and in period of age in our life, this exposure might trigger or might not trigger, and it's getting this characterization in perspective also of our pattern of life, or whether you are more prone to be inside, indoor, or you're more outdoor, and understanding that dynamics. So I think overall, it was across the 5 groups, there's quite a lot of nice overlapping ideas. And if I think what we will be looking at, and if you are interested, please do get in touch. But I think this compendium or this, at least somewhere, I don't know if it will be a book chapter, a white paper, or central database, but trying to already identify methods and technologies, and I think identifying what you are looking for, what the endpoint, because obviously if you look for pathogens, you might be more on bacteria, etc. But if some people are very much only on fungi, some people might say, well, I work on endotoxin or what matters, what are you looking for? And perhaps we should have these summarized somewhere where you can find the information and getting this decision making on what might suit your needs and address your endpoint. And also we had a quite good discussion with, and this is where we need to link with our health people. You know, we were asking them, giving you CFU per ml or cell per ml for my qPCR, that help you to understand how the health issues affect the patient, etc. And there is helpful, but sometimes it's no straight connection. So it's how we can provide data, but also making meaningful context for tackling the bioexposure issues. So I think this would be quite useful. And what to target, what shall we measure? On this good point, I'm bearing in mind the time, we are 2 minutes now over the time. If there's anyone want to add any final comments, please do. And you also please do send us an email or feedback. What we will do, we will summarize the discussion of today on the workshop, and there will be a release on the website. So we will send to all of you an email, and then we intend to develop some activities. And if you want to take part and join, we will be very pleased, and we can certainly initiate few initiatives from what we've been discussing today. And also a part of the network today was very much on sampling and characterization, but we got 4 themes. So on the first theme, Sean is there, is going to share with our colleague Jan Kolbeck, where they're really trying to look at the broader picture and the dynamics, and how that we need to get this holistic approach also. We got Emma, she's co-sharing also with our colleague from Plymouth and Oxford University with the themes 3. And they are looking much more at the health impact and how we translate our measurement to indication on how that might affect our well-being, our vulnerability, our immune response. And then we got also colleagues more on the policy context. So you will see we are going to advertise other thematic workshops. You don't have to go all of them, but if you are interested, the idea is always trying to articulate some needs and how we can move forward these different aspects. Anyone want to have comments, final words? If it's not, I would like also to thank you, thank Carla and Rob for the time to present and give their thought, and also thank everybody today for your time and your comments and feedback. That was really beneficial and helpful for us.
Thanks, Fred.
Thank you.
Well, on those, wish you a good afternoon, and hopefully we will see you soon on our future event and follow up. Thank you very much.
Thanks, bye.
Bye.